So the next speaker is actually an invited speaker. Susan Poteet have been courting Andrew Sewell for the last 2 years to give a talk. The first time I heard about Andy's work was actually from a patient who was like, bring me this article about T cells that cure everyone, any cancer, so to say. But he's going to talk about that particular work, but you will find him to have a very refreshing look at T cell biology and targets. I think it's going to be very, very interesting. So, Andrew Sewell, thank you so much for visiting us and telling us about your work.
Oh, thank you very much for having me. I'd really like to thank the organisers, particularly Brian, but of course Susan, who I couldn't say no to. So I get invited to these meetings all the time and just say no, but Susan's not someone you are able to say no to. So, yeah. These guys are examples of what great advocates you have. I think I'm on speaker, aren't I? Yeah, I can be heard. The guy in the mustard top at the back said he could control me from the back of the room. I said, so if this goes wrong, it's his fault. And please don't tell my wife you can do that. But your advocates are fantastic, I have to say. So I couldn't really say no. So I hope I do a good job after all of this. And I'm not going to talk about the HLA agnostic T cell work, but if you wanted, I could go through some of that in lunch break or whatever we work out. So we'll start here. It's nice to see all these people who work on kidney cancer in 3 dimensions. It's nice again. I'm going to start off by saying that I don't actually work on kidney cancer, so I'm a fraud. I shouldn't really be here. So I do apologize, although I do have a little bit of kidney cancer in there, I realized, without knowing. So I'll definitely point that out. So I'm coming at you from Cardiff, which is the capital of Wales. So Wales is one of the 4 United Kingdoms that make up the UK. It's the purple one on the left there, and it has the coolest flag in the world in every vote they ever do. And you can see Cardiff's kind of down the bottom there. Got to get fed up of explaining that when you move to Wales. So I made this when I first got there. This is the easy example for everybody. Wales is the red bit. The blue bit is not Wales, okay? That's done. Now you know where I'm coming from. So in Wales, what I do is I work on these cells called T cells, and they're fascinating cells because they orchestrate immunity, and we all know through the COVID pandemic that they protect us against infection. And they're more important than I think that people realize in doing that. What we're here for today is we know that they can eliminate cancer. So I'm going to tell you a little bit about that. They're also essential for vaccination. So don't believe these people who say antibodies is everything about vaccination, because you cannot get a good antibody without first having a good CD4 T cell response. Without that, antibodies are useful. So T cells win hands down. Back in the day, they just used to be called lymphocytes. And I used to go to the lymphocyte activation meeting. And our cry was T cells are terrific and B cells are boring. We've already done B cells. T cells are definitely where it's at. So they do a lot of good. They also do a lot of bad. We've got that listed here. They're the root cause of pretty much all autoimmune disease. And this is something we work on quite a lot in the lab. They're also responsible for some allergic reactions. But as we start to look at it, it looks like they're responsible for most allergic reactions. And they're responsible for the acute rejection of transplanted organs. So if you're going to work on something, work on T cells. This is 80+% of human pathology here. So we work on all of these aspects. And the key to it all, and Hootie did a great intro to these, is this T cell receptor, that thing on the left, binding to this thing called, I would say, a ligand. You'd probably call it a ligand, whatever. Same thing, tomato, all of that. Yeah, that thing. Hootie did well there. Right, so you're a difficult audience to come across because basically I realize there's patients here and there's people with no expertise all the way through to people who are probably more expert than I am. So I thought I'd introduce them, but Hootie did a really good job of this. One of the types of T cells that are my favorite are the killer T cells. These are CD8 T cells. And they have some superpowers that I'm going to tell you about. And one of the main superpowers they have, and they're the only thing with this in your body, is they have X-ray vision. They can see inside other cells. And the way this works is the proteins inside the cell, little bits of them are presented at the cell surface in these molecular cradles, generally called MHC, but in a human, they're human leukocyte antigen, HLA molecules. So you can see all of those little bits on the top of that cell. You can tell I like to point at things, but I don't have a pointer. It's doing my head in. And the T cell receptor, this little barcoded thing that Hootie was talking about, basically does that recognition. And we all have about 100 million different T cell receptors. And to put this into context, and I think people don't realize, is that identical twins don't have the same T cell receptors because of the way they're made. And this is how you can end up with them being discordant for autoimmune diseases and such things. So normally only self-proteins are in the cell, so the T cell should ignore that cell. In this case, we've got a virus inside the cell. You'll see it's making some viral protein. So viral peptides will get out on the surface, and then the T cell can recognize them and activate them. Now, we now know that T cells can also see dysregulated gene expression inside cancers, because cancers express different proteins than normal cells, and they can see the mutations that might be occurring within the cancer. So to use the supernova analogy a little bit more, the reason killer T cells are so good is because they can actually nuke the target. So they're a very blunt weapon, but when they activate and see a virus inside the cell, or note the cell is cancer, they just destroy it and all of its content. So I do a lot of things, but I'm increasingly being forced by money and things to focus on cancer. And we ask a very simple question in terms of cancer, and that is, well, what do successful T cells target? In order to do that, we look at three main cohorts. We look at patients who've been cured of cancer, complete remissions from tumor-infiltrating lymphocyte therapy. That's the one I'm going to tell you about today. It's very straightforward. We put the T cells in, they work. So we know what we put in, and we can just follow it. And we're going to dissect some of that today. We also look at checkpoint inhibitor cures or complete remissions, and we look at natural cancer survivors. So we'll focus on the TIL today and do what Hootie wanted us to do. We'll tell you how we find what a T cell recognizes. So TIL therapy, for those that don't know, is basically taking the T cells that reside in a solid tumor, growing them up ex vivo in the lab, and then putting them back into the patient. Now, I still like our original cohort. Now, this is done at the Center for Cancer Immunotherapy in Copenhagen. They've just run a phase 3 trial in melanoma, hundreds of patients. The original cohort is over 10 years old, and there weren't any other immunotherapies around in those days, and it was very clean. So what they did, they take 1 gram of melanoma tissue, grow up the T cells in it, and 10 to the 10 was the go, no go. If they get 10 to the 10, that's a 1 and 10 zeros, big number of T cells, we'll see in a minute. They put them back into the patient after some preconditioning. And even in this original cohort, the 33 that were treated, 7 underwent complete durable remission over a decade ago. All 7 are still alive. You know, they were, in effect, cured. And to give you an idea how sick these patients were, because they're stage 4 melanoma, 15% takes 4 weeks to grow the TIL, 15% of them died or were too ill to actually treat. So these were very sick patients. And when you see how remarkable it is, the size of the tumors that get taken from some of these people by these, it is astonishing. So we get over 20% lasting complete durable remission. So we're really studying those patients. So when I first saw this, I said to Inga, I said, I'll tell you how you've cured your patients. Because the reason I like to work with Copenhagen is they have fantastic samples. And the unique thing they have is they have the autologous tumor line from the patients. So we just take those. So we've got the blood before treatment, which is actually the most valuable sample because we didn't take enough to begin with because we didn't realize that would be nice. You have the TIL infusion product. There's lots of that stored, so we know what went into the patients. And then we have the PBMC or the blood after complete remission. And these patients are incredibly compliant. Once you've cured someone of an incurable cancer, if you want a sample, they'll come up and give you one so you can get it. So you can tell how old this is. So this is really what we're doing. That sort of picture on this that I can't point to is a wave bag. We don't do it that way anymore, but that shows how old this trial was. So that's the TIL infusion product growing up in the lab. And then we take that, and I guess in the flow cytometry plots, we're using a technique which is basically using fluorescence and molecules that bind to other molecules to actually find the T cells within the TIL infusion product or within the PBMC that activate in response to the autologous tumor. So we drop the tumor in and we can find the T cells that activate. So our research question is quite simple. Okay, these T cells cured this patient, so what do they see? And I'll tell you now, they don't see anything anyone's ever worked on before across the board for all the ones we've looked at. Now, I was really disappointed when we first did this because we sequenced those barcoded T cell receptors. And in general, within the TIL, there's a wide range where we see 200 to 300 different T cell receptors responding to the autologous tumor. And I just say, Inga, that's too much for us to work with. So thankfully, when we then go in and have a look in the blood, so the PBMC after complete remission, we often see this kind of picture, where sometimes 2, but generally 1 T cell receptor comes out and dominates the response to tumor. Now, everyone who knows anything about this at the moment is thinking, yeah, that's going to be a neoantigen, but wait for it, because it's not. So we ask, what are those recognized? Sometimes they do see neoantigens, but more often than not, they don't. And what they see are new shared antigens that occur in other cancers. And I'll eat my hat if they don't also occur in kidney cancer. In fact, I think we know some of them do. So I very much see this as sorting the wheat from the chaff. You know, we don't know what T-cell, what targets are the right ones, but this is a way to, you know, build a hypothesis to say this is a good thing to target. And indeed, in some of these patients, we've seen the dominant clonotype targeting the same antigen. The chances of that, I think are very small. I like this analogy of wheat and chaff because my long-term aim is to grow the wheat. I'm going to vaccinate. Now I know what T-cells might work, and I actually have money to do that. It's going to be a slow process to try and vaccinate, to generate these types of T-cells in those patients. So this is the bit Hootie wants to know. How do we find the targets? This is a bit complicated. I've got that red box in there because it's going to blow people's minds who aren't necessarily scientific, but just slightly to talk you through it, and then I'm going to run you through an example. So first we find the T-cells. Second, we find the T-cell receptors. We developed technology to do that ourselves, but you can now do that commercially. We then, in effect, build the T-cell. So we take, say, a killer T-cell from someone else, we knock out the TCR genes and put in, with a lentivirus, the gene, the TCR that we want. So we then build that T-cell receptor clone. So in number 4 there, the one we're going to follow today is a T-cell receptor called MEL8. That's the one we're going to follow forward. Because when we've built them, we don't just want the ones that see melanoma. We want the ones that see breast cancer, lung cancer, and all cancers. And they exist in there. So MEL8 is one of these, and we're going to go through and see how that works. So we'd say that's worth putting through our pipeline. Our pipeline, because CD8 T cells see small peptides, and they can be of different lengths. First off, we need to know what length, and they tend to have a length specificity. So we find out what length, and I'll show you an example in a minute, and then we screen them with a max length combinatorial peptide library, and then make our predictions. This was computationally heavy. We now do it on graphics processing units, and beat the university mainframe by hundreds of fold. So this process I've just called Pantigen, because it finds cancer antigens. So here's an example. So this is melanoma stage 4 patient number 24. In panel A, we can see the TIL infusion product from this patient over 24 hours was very good at killing the autologous tumor. It also kills other melanomas in panel B, providing they have the most common HLA in the population, HLA-A2, which immediately says, okay, most of these TIL are not a neoantigen, because you're not likely to find the same neoantigen in all these other cancers. They don't see A2 negative melanomas, which again says that most of the response looks like being through A2. In panel C, we can use an A2 blocking antibody and then show that it is A2. But what I really want to focus on is what we do. Because we have the autologous tumor here, what we can do across the board with these patients is we can knock out individual HLA heavy chains using CRISPR from the autologous tumor. So in that red box there, panel D, the top is the response of the TIL infusion product used to successfully treat this patient to the wild type tumor. The one below, we've knocked out HLA-A2. So you can see most of the response in this patient is through A2, and it's a shared response. And indeed, it sees a whole bunch of other cancers, but we'll do that in a minute. We'll come back to that now. So the TIL infusion product sees a wide range of different cancer types only through A2. So the way we do that is if it's an A2 tumor, we knock out A2 and we remove recognition. With a non-A2 tumor, we can just transfuse it with A2 and then it would be recognized. Now, the dominant clonotype in this patient is a melin A. It's a very common melanoma antigen. It's overexpressed in melanomas. It's probably one of the most worked on cancer antigens out there. So we know that the dominant clonotype, MEL8, sees melin A, but it also sees other tumors. For instance, whatever we've got there, ovarian and such like. Don't have kidney on there, but I'm sure they will see. They will see kidney in the end, I think, you know. So, so melin A isn't expressed in those particular tumors. So we were a bit confused by that. So then we CRISPR'd out the melin A gene from the autologous tumor. So normal, and they're represented by the triangle and the square, normal melin A specific T cell clones now no longer see the autologous tumor, but the MEL8 clone does still see the autologous tumor. So killing of other cancers is A2 restricted. The The MEL8 clone also kills other cancers that don't express melan A, even though we know it's a melan A-specific T cell. The MEL8 recognizes wild-type tumor even when we knock the melan A gene out. So you don't have to be Sherlock Holmes to work out, well, this MEL8 clonotype must see another widely shared tumor-associated antigen. But what? So we're going to run this through the Cantigen protocol. So this is a core technology in our lab, which we call a combinatorial peptide library, which we lay out in positional scanning format. This is the point where I lose everybody, but I'm going to try not to. See that little 0 and X's in the middle? That's a representation of a 10 amino acid long library, and it's arrayed as 200 sublibraries. So sublibrary 1 has an alanine fixed in position 1, and an equimolar mix of the other proteogenic amino acids in the other positions when we make it. Sublibrary 2 has a cysteine fixed at position 1. Because this is a 10-mer, we go all the way down to sublibrary 200, and it's alphabetical. That's a Y, tyrosine, at position 10 in an equimolar mix. So 200 sublibraries in there. T cells see these, tell us what they like. So each sublibrary there contains over 3 times 10 to the 11 peptides. Big numbers. They're any one peptide, be at tiny concentrations, but T cells see them. So if you want the background to this, I'm just putting in this paper. It's not a cancer paper, where we did a similar technology for an insulin-specific T cell. But if you're interested in that, I'd refer you to that to sort of look at the algorithms and the type of things that we're doing. Coming back to cancer, Cantigen is now available as a web tool. It's password entry at the moment because we haven't really published it, and I'll show you what it can do in a minute. But what it does is it takes the self-protein database, and then within that silos a sub-database for scoring purposes. And obviously, to score highly in this, you need to be a validated cancer-associated protein. And it works well if you do it that way. If you go into sort of mRNAs, it seems to get less good at doing it, because I think some of those aren't particularly real. So, that done, what does our new broadly tumoricidal T cell clone MEL8 recognize on the surface of most A2 positive tumors? So here's the actual raw data for this MEL8. So the sizing scan says it likes a 10 amino acid long peptide. So the other there are those 200 sublibraries tested, 10 amino acids long on this clone at 100 microgram per mil, which is the highest concentration we use them at. We can immediately see that only a glycine really shows in position 6. If we back off and do 10 microgram per mil, the next thing we see it likes is an isoleucine at position 7. But regardless of this, we just take these data and we just run them down the Cantigen pipeline and say, okay, what does this T cell see? So here's the data. We'll talk you through it. Panel A is just a recap. The clone sees the autologous tumor. It doesn't see it when we knock out A2. We know it likes a 10-mer peptide in panel B. Panel C is the data I just showed you, but as a heat map. But on it, we've marked the melan A peptide that we already know that this T cell clone sees. So the key panel here is panel D. This is the raw output of Cantigen scored. So we're going to concentrate on the top 4 there. Reassuringly, the second best prediction Cantigen says that this one sees is actually the EAA melan A peptide. So it's reassuring to find what you know it already sees. Funnily enough, the top one wasn't seen, but we're going to focus on number 3 and 4, which are peptides from bone marrow stromal antigen 2 and insulin-like growth factor 2 mRNA binding protein 2. And this TCR sees all of those peptides. And indeed, on the bottom there, this is tetramer staining. The MEL8, which you can sort of see on the left, stains with all 3 tetramers. We then went back to the TIL from patient 24 that cleared their melanoma and found 3 other clones, which you can see there, that also have this phenomenon, that see these 3 different epitopes, and they actually recognize them all. What we then had to do was find a tumor cell that doesn't express any of these epitopes. Every cell line you use in the lab is pretty much a tumor cell, except if you're using primary cells, which are a pain because they don't grow for very long. We struggled to do this, particularly for IMP2, but MALT3 cells don't express any of these. They don't express A2 either. So if we put in A2 and melan A, or A2 and IMP2, or A2 and BST2, these are now seen. This is important because it shows that these epitopes are genuinely processed and presented. So we've got these in other flavors as well, as I'll show in a minute. But for want of a better term, I don't think anybody's come up with this before, but I've basically termed these multi-pronged T cells. So a multi-pronged T cell sees its disease target through different peptide epitopes on the surface of that same target. So to run you through that again, on the left, normally what you'd see is a T cell seeing one peptide, and then it would stain with one tetrameric, multimeric form of that peptide. Whereas on the right, the multi-pronged T cell is basically seeing 3 different epitopes in this case, and stains with all 3 reagents. So we've got one T cell receptor recognizing multiple cancer-specific peptides. And indeed, in doing this, in this one example alone, we've found 2 new, widely expressed tumor-associated antigens in BSD2 and IMP2. So on the left, sorry, I'm right-left dyslexic, so it's hard for me, this, is the expression of these 3 proteins in the autologous tumor that was cleared. And you see all 3 are expressed at really high levels, even IMP2, which is the lowest, that's still a high expression level. And you can see across, and we've even got some kidney cancers in there, cell lines. I don't know what they are, but I'm sure that the audience can tell me what they are. But basically, pretty much all tumors express one of these genes at high level, one of these proteins at high level, and most of them express both of them at that kind of level. So then you're going to ask me, well, Andy, how does one T cell receptor respond to 3 different antigens? And the answer is, and this is real, now, we actually did this. Crystallized it, looked at it with X-rays. These are the structures of one TCR. And as you can see, although the sequences of the epitopes are different, on the right there at the top, the amino acids are different. They're not really different at position 6 and 7, and position 4 is the other hotspot. So this TCR, and that you've got the epitope sequences there of those 3, it's looking for a 10 amino acid peptide. It must anchor to A2. So in this case, that's positions 2 and 10 must allow that. And then have a glycine and isoleucine at position 6 or 7, and it prefers no side chain at position 4. I think the message here is clear. Multi-pronged T cells kick ass. I just wanted to sort of drive it home with that kind of thing. Panel A, and this is something I don't fully understand yet, but it definitely is reproducible. Having the 3 epitopes there seems to be somehow they cooperate, and I don't know how that works. But what we really wanted to do was compare a multi-pronged TCR to a regular TCR. So the one we picked there was DMF4, which was the first ever TCR-T trial from the Rosenberg lab. In melanoma, it's a melanase-specific TCR, published in Science. You can sort of see it there, top right. DMF4, bottom left, doesn't see these other 2 epitopes. And to cut the long story short, in head-to-head comparisons, if we put the multi-prong that sees multiple epitopes in against the single one, the multi-prong wins every time. It's better at seeing melanomas, and of course that one actually sees other tumors, whereas the others don't. So the big question you should be asking me now is, look, Andy, it's all very well, but is it safe in vivo? We've got the data. It is safe in vivo. Let's go back to our original patient 24. They were treated with 1.1 times 10 to the 11 TIL. Yeah? That's the cell pellet, Mark O'Donoghue holding it there, that went into the patient. 78 mils. Wouldn't fit in a Falcon tube for those of you in the lab. For the rest of you, look it up on Google, 78 mils. It's about the volume of 2 golf balls. So this patient was treated with 2 golf balls worth of T cells. And we have them, got lots of them stored. So then we stain them across the board for different A2 specific epitopes. There's not much in there seeing HIV. There's not much in there seeing insulin. But we can see a whole bunch of them that see MELAN-A, IMP2, and BSD2. And when we sequence the TCRs that stain with those, they're the ones I've been telling you about. So we know that this patient received billions of activated T cells with this specificity, and they're still alive 10 years later. And you can still find the Ts, not at this sort of level, but still find them there now. We've also found this type of T cell expanded in other complete remission patients. I don't have time to show you those data. But I thought what I would show you is data from soluble cancers, because there, of course, when we take a blood sample, most of the cells in it, when they present, is actually the cancer. So you've got the cancer directly ex vivo and can do things. This is the most remarkable we've seen. This ME91 is an AML patient. When they pitched up in Cardiff about 3.5 years ago, we had a bit of sample left, so we stained them. One just didn't activate them or anything. 1 in 50 of their T cells stain with an IMP2 tetramer. We didn't have the BSD2, but they did stain with a MELAN-A as well. And you'll see in the bar chart there, they kill the autologous tumor. So our other example now on the right is a patient with chronic lymphocytic leukemia. They had some IMP2 T cells, but it wasn't really enough to do anything with, so we stimulated them out for 10 days there. It's a slight difference here. But those T cells are pretty much a clone. They kill the autologous CLL cells from the patient direct out the arm. And of course, when these things are in patients, we already kind of know they're not seeing healthy tissue, but we've done a whole bunch of healthy tissues down the bottom there. They're not recognized, even though they're A2 positive, but they are when we add the IMP2 peptide. And this is a collaboration with Oliver Ottman. So whoever's done the work, I've been putting that on through the slides. So really to conclude, I've still got 5 minutes left, which is good. Persistent dominant cancer-specific HLA-restricted T-cell clonotypes following successful immunotherapy need not recognize neoantigens. Now, I'm not saying they don't, sometimes they do, but they don't have to. I think more often than not, they don't. They can recognize new shared antigens. In actual fact, the antigens we're finding, no one else is working on. And sometimes within those TIL, whether they be Wilms tumor or GP100, they're in there. They're not the ones that expand out in the complete remission patients. And finally, these T cells can recognize multiple tumor-associated antigens simultaneously through a single T cell receptor. So the example I've shown you today is one that sees 3. It's the red one at the top on the left there. We also, even through A2, have examples of ones that see 2 or one that sees more than 3. We think 4, could be 5. Haven't actually managed to knock them all out yet. We've knocked 3 of them out and the cancer's already still seen through A2. So they're not BST2, IMP2, that sort of series. They're different ones like this. So the real message there is this is not a curio. There appear to be other flavors of these types of T cells in patients who clear their solid cancer. And obviously, we can't do that at the moment, but we'd like to say that having these kind of things is a good thing. We've definitely got data that indicates that, but it's not statistically significant. So multi-pronged T cells are rarer T cells that recognize multiple different peptides on the surface of the same diseased cell. I don't think there's any examples of this really out there, but they're more difficult to escape from. So if you had 3 T cells seeing 3 antigens, you escape from any one, you have a Darwinian selective advantage, and you'll go down that route. If you lose one when you've got against a TCR that sees all 3, you have no advantage. Clearly, you could lose the HLA, but that's another story. They're more sensitive. It makes sense. Anyone who works in T cells like Hootie knows when you add more peptide, you get a better response. So the fact that they're seeing multiple different peptides seems to make them more sensitive to tumor. And then you need less T cells, in a sort of head-to-head. And ultimately, because they're seeing multiple different antigens, then more tumor types are recognized because they're more likely to express one or more of these antigens. So Brian asked me to finish up and say, talk about the future. So the future, number one, and I don't know if I'm going to get time for this, but I should start working on kidney cancer, really, shouldn't I? Now I've been invited to you. And you bet your life Susan's going to work on me so hard for that. So we can't rule it out because we know her powers of persuasion. Next generation TCR-T therapies. So these are when you put TCRs into the patient's T cells and then put the T cells back into the patient, like CAR-T, but with T cell receptors, should really try and replicate known successes using successful HLA-restricted, and I haven't talked about the HLA-agnostic T cells today, but we have a bunch of those as well that don't see MR1 and see lots of different tumors, that recognize cancer in new ways. I mean, to me, this is not rocket science. Let's take what's already worked and try it and reproduce it in other patients. And one thing I really want to get at you here is what we're finding here by looking at these types of patients, I think we're finding cancer's Achilles heels, because some of these antigens we can't even CRISPR out of any tumor line. They just stop growing. And I don't know whether that's true in vivo, but it's just a way of homing in. So at the moment, all we do is take square pegs and bang them into round holes and say, yeah, this is a good antigen, we'll do that. I think it'd be far better in future to go ahead and say, let's have a look at what's already worked and see if we reproduce that, whether it would work again. And then as we have it there, kick cancer's butt. That's all, folks. Look at that, on time. There you go. Sorry if it was a little fat rush there, but we've got lunch now and I can go through things. These are a lot of the faces. This is obviously, I think it's a photo from February 2020. After that, we're all 6 feet apart wearing masks, but we got us all together there. We are now hiring. all those with mortarboards are PhD students who are the general lot that do this work. In terms of the real acknowledgements, Barbara Sommelier in Cardiff does a lot of the mathematics for us. The rest of them listed there on the left is my group. Now, before you think I have a group of 30 people, the current members are only the ones with dots. This is done in series through 3 years our PhD students get, going, training them up, getting them, and they go off and get fantastic jobs, and we never see them again, unfortunately. I should thank Hematology, we did the solid cancer with. We couldn't have done any of this without the Centre for Cancer Immunotherapy in Denmark, and Inga and Marco are brilliant and fantastic collaborators. A lot of our sort of algorithms and sort of stuff we went into is through the University of Warwick with the guys listed there. And our funding, I really want to thank the Wellcome Trust, who funded me now for, I think, 26 years straight. They are brilliant, and they've been really good with me. We did just land a grant as a Cancer Research UK National Cancer Institute Grand Challenge. So we went in on this for childhood cancers with an HLA agnostic approach, possibly through CAR-T, $25 million. So we are hiring if anybody's got any money. That's a consortia. Unfortunately, I don't get $25 million because most of it's going for the clinical trials that we hope to develop. We do get money in the lab from Anara Bio, really through the MR1 and then some other projects, and a whole bunch of sort of local in Wales cancer charities funders. Thank you very much.
Do you want to do questions or?
0-0.
Well, that was
To the second.
yeah, no, that was outstanding. Questions? Lisa.
It's the guy in the mustard jumper that did it. He's controlling it.
Too restricted,
Right.
HLA typing.
So,
how common or how rare is it?
so that's a very good question. It's a fantastic molecule. Now you're going to get me off on some nerdy side train. HLA-A2 came into the human population through Neanderthals. So they were already in Europe, they'd already developed it, and when Homo sapiens left Africa, we interbreeded with them, whatever. Where I'm from is 53% of the population in South Wales. Generally in the UK, it's 45%. It is the most common HLA in the human population. The second most common is HLA-2402, which came in from Denisovians, who were there when, so what we've done is, basically, they were out of Africa before us, evolved HLA that were really good with whatever the local infections were, and then we inherited them by interbreeding. So when people say, we're 4% Neanderthal, or people in, Tibet are 6% Denisovians, those percentages is basically the HLA, is the main bit of it. So very common, but very good question. Thank you.
Does it cost the blood-brain barrier?
T cells, yeah. They can.
Jim?
So, couple of questions. One is, so these T cells that with just one T cell receptor recognize several different epitopes.
Yes.
I suppose the epitopes are fairly well related to each other.
In this case, yes. The key binding hotspots are conserved between the epitopes.
Okay. Could one use informatics to try to predict potential such epitopes?
You could. I just do experiments. I quite like, call me old-fashioned, but I just like to do experiments.
Okay.
And I think if you did enough of these, and obviously this is things that people are thinking of, you could screen, because we can do this robotically in terms of liquid handling robotics, screening T cell receptors. If you screened enough receptors, certainly if you screened 1 million, maybe if you screened 100,000 receptors and then built that on top of what's already known, then I think you could start to make those predictions.
Right. And so my last point is, so you mentioned that, of course, if you just have a T cell that is targeting a single antigen or a single ligand, escape is a lot more likely than if you have 2 or 3. So could you speculate, what do you think is the frequency of having such T cells in patients that have durable, complete responses to immune checkpoint inhibitors? I realize it's a speculation,
Yeah.
but I mean, what would you speculate? Do you think this is going to be the majority? Is going to be sort of the theme, or not? Or what are your thoughts?
My gut feeling is it is relevant, but again, I don't have statistically significant data in that. But the data do appear to be black and white. So let's take melanoma patients who are A2, who are cured, complete remissions, and the ones that just progress at the other end. You know, maybe a quarter of them, a third of them just progress as they would have done anyway. And let's, for instance, stain with an IMP2 tetramer. All the, in our hands, small cohort, but all the ones who are complete remission stain, and none of the ones who progressed had that particular antigen in there. So I think you can do these kind of things. I just don't have the bandwidth. It's frustrating. So, I mean, but once you've found the antigens, the beauty is you can make the tetramers and then you can go looking for these things. So when we've done this in other complete remission patients, now it's quite quick, once you know the answer, to go in and find them, and we find them. But that doesn't mean, unfortunately, we haven't got a whole bunch of patients there that we can't get blood retrospectively from the ones that didn't survive. So it's possible that they were there then. But the nice thing that I like is it says to me these types of T cells are safe in vivo because we've seen them in multiple individuals. So if they were attacking any self tissue, you would see the pathology.
Dr. Sewell, one of the questions on the chat was whether myeloid T cells can be kept off on the shelf, or do they have to be assembled during autologous cells?
Well, that's the way things are going at the moment, isn't it? So basically, I think at the moment, let's remember that cell therapy is very expensive. And this is why my long-term aim is really vaccination. I can pinpoint these types of T cells, not only with a peptide, but with a non-peptide. I can induce them, in effect, with a drug. Because all T cells do is they see non-self shapes bound to self-HLA. And those molecular shapes do not have to be peptides. And we've got non-peptide things that will induce them. So in an A2 transgenic mouse, I can feed the mouse the drug and induce the T cells without any adjuvant or anything. We did this with D-amino acids. The only thing we've published wasn't great with the D-amino acids, but you can look it up. It's in JCI. And we used a D-amino acid peptide, completely different sequence. And we could protect A2 mice, so humanized mice, against a lethal challenge with influenza, simply by feeding them the D-amino acid, because they don't digest it. So this is the beauty of these kind of chemistries. They're more stable type of thing. So what was the question? I went off on a tangent.
No, it's just about whether they can be off the shelf or
you can. Well, so what people are doing now, I guess if people aren't in all this field, is really even using pluripotent stem cells. So then you take your stem cell, you do your anything you want with it. You take out the HLA, you can add in anything you want, and you can put a T cell receptor in there, and then you turn them into T cells. And I've seen sort of 60-liter cultures of these things that certainly in vitro seem to look like a regular T cell. I'm not sure. The NK cells, obviously fake therapeutics, have put those into people. But so the way to go is to be putting these kind of things. If you ask me about off-the-shelf therapy, why not use an HLA agnostic receptor from a patient who's cured their tumor and it was the dominant clonotype? They are incredibly rare, but I'll tell you they exist and we have them. So I would be more inclined, because then you could just treat anyone regardless of what their HLA was in the future. I'm not saying it's going to work, but it'd be great experiments to do.
Dr. Atkins.
What do we know about how these novels are expressed?
Sorry. What do we know about how these novel shared antigens are expressed in tumors?
Well, there's obviously I just showed you one set. So in melanate, there's been lots of work done. Melanate is expressed at low levels in melanocytes, and it's overexpressed in melanomas. So that one we know. BST2 seems to be expressed at reasonably high levels in most tumors. The nice thing about BST2 is it's cell surface as well. Now, the kind of questions I was expecting to get here is certainly there's literature out there to suggest that some of these antigens may be expressed in some healthy tissues. Yeah, and that's an interesting thing, because in effect, we've done the experiment that you couldn't really do, to be honest, by doing the TIL therapy. You put these kind of things in. I think that's a really interesting question, you know, are they genuinely expressed in any healthy tissue? And if they are, well, why aren't those tissues seen? I don't know the
Well,
answer to
I
that.
guess I was driving at if you're using vaccination or off-the-shelf T cells, you've got to make sure that something's expressed by the tumor. And so you've got to identify, what's expressed at the tumor. Or
We
figure
know
out
it's expressed, and
how to
obviously
induce it to be expressed.
So we've got single specificity BSD2 into melanin A T cells. So they're not the multi-pronged sort. So normally if you give the peptide, you get the normal T cell you'd expect. They kill the tumor. Then we can CRISPR out, for instance, the BSD2, and then they don't kill the tumor. So we know, and bear in mind, these are all in vitro tumor lines. We haven't done a whole lot yet with primary tumors, because it's just a bit more difficult to work with.
Tony?
Last question.
Fantastic talk, of course. My question is, you mentioned about the long-term survivor, the one who cured. You go back to the lab when you already collected, or these patients just now they come and they have some circulating T cells that have anti-tumor activity, and you increase them. I don't know the difference.
All of the above. But this is why I kind of like the Copenhagen cohort, because I can cherry pick the ones where we've got the autologous tumor. And we can go back to a patient who was treated, 2 years ago, 10 years ago, or whatever. And obviously, if they're a complete remission patient, they're still around, and then we can start having a look at those.
And final question. So, we have a lot of people, let's say, discouraging us. Sometimes young researchers are going toward cell biology. No, the disease is busy, whatever. So, a lot of attempts to look after NK cells. Same thing. You think different biology, I get it, but do you think the same approaches, what you're doing could be applied to another cellular therapy like the NK cell, which are gaining some ground?
Yeah, as I said at the beginning, I like T cells. And the reason I like T cells, in large part, is that they are incredibly sensitive. And this is true of the HLA agnostics, which actually makes the biochemistry quite difficult. But a T cell can see 5 molecules. What else in biology can do that? A moth antennae can maybe see 1 or 2. You're really at the limits. NK cells just, macrophages, all of they're not even close. So fine if you're going to look at, a lineage-specific marker like CD19 where there's 100,000 copies, but that's not what these T cells so far appear to be seeing.
Speaking of CD19, the context of that literature, I've seen some nice data suggesting that, thus responses or, for patients with lower volume at whole phenomenon of effector to target ratio. Do you presume that with your strategy, we would see the same, or do we see responses irrespective of volume of big bulky tumors?
If you actually look at these patients who are treated with TIL, with the melanomas, some of those, I think it might have been patient 24. I get confused which one's which, but had I would say, not quite the size of an American football, but, something like that round the spine, right round the spine, inoperable, just leave that there. Within 2 weeks, it had all collapsed. And all you see is the sort of skin and the backbone. It's incredible kind of necrosis going in. So you have to ask the clinicians, because I don't treat the patients. But from what I've seen, 2 weeks after you've treated them, you can almost say, that this is a good one, because the tumors just start to collapse. So even the large, bulky tumors. This is the problem with CAR T. We can develop it, and I know there are developments going on at the moment that maybe I shouldn't be telling you about, so I won't. But a T cell, in my lab, I've watched a T cell crawl over 5 cancer cells to kill the next one. Because they're light touch, particularly tumor-specific T cell receptors, KDs, sort of 50, 100 micromolars. Antibodies aren't anything like that. They're basically superglue. So you take a T cell and you cover it with superglue for tumor. And guess what happens when you put them into, say, an animal with them, or probably unlikely a human? The first tumor cell it sees is the last one, because it's just stuck. So they don't actually penetrate in there. So real T cells, we know through TIL, certainly in some tumors, can actually have that ability because it's part of their job, I think. Whereas what you're doing with a CAR T cell, well, I like T cells, I like real T cells. So this is when I go to a lot of car meetings, and I used to upset them back in the day, because the T cell is basically the thoroughbred racehorse, the T cell receptor. It is the Formula One car. We don't know anything really about how that works. So what we're now doing with a CAR T is breaking down your Formula One Ferrari and building a bicycle. And that's literally what we're doing. You know, we're just putting a bit, oh, well, the wheels go there type of thing. What I have seen, and I think I can tell you about that, so Robbie Manzana in Stanford has cut out the middleman, because he's got this on a pre-publication website now, and I really like the data. So all of these signaling domains, everybody's, you know, do you need, you know, CD28, Zeta, blah, 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 and everything, they're all there to recruit scaffolds like ZAP70 or LAT. The best cars cut out the middleman. You just put antibody, membrane spanning domain, and LAT and ZAP70. They work much better than any of the current generation of cars. It's not rocket science, in effect, once again. So, and I now think certainly Rob is getting very clever in terms of specificity and sensitivity now. So we are learning. So I think I don't know what generation cars are on, but by the 12th generation, they might be okay. So I'll give them that.
Well, there. Last question.
Yeah, fantastic talk. We often see mixed responses when we treat patients, like lung mets getting smaller but progress in the liver or bone. But when we isolate those cells, we end up getting tissue from one location and then produce. Like those T cell property are shared across the different site of disease, like any challenges or any barriers down the road?
Oh, I think there are going to be lots of barriers. So I'm not presenting this as the answer, because I think what you're going to need are sort of combination therapies. But you know, if you're going to do something with a T cell and a receptor, then my hypothesis is these are the ones. You know, go with the ones that have already worked and do things better. But I think the tissue location is very important. I mean, something like the liver isn't very friendly for T cells. So yeah, you may have to get kind of clever to get around those things. And of course, nobody's mentioned the dreaded microenvironment. But of course, with these off-the-shelf things now, you can do everything with your stem cells to begin with. So, and I mean, Carl June locally, just over there, has put them into patients where he's knocked out PD-1, knocked out CTLA-4, because all the toxicities associated with those is because you're basically taking the brakes off all your T cells. But if you can engineer your T cells without the brakes, then only those ones are affected. So I think in the future, that's a possible way to go. It's a fun time to do cancer research, I think. Yeah.
Outstanding. Well, thank you so much. This comes to the end of the session. There's lunch outside. Now, I think, go ahead.
Yeah, we're going to invite anyone. We're going to block that back door open. People want to come and go and bring their lunch in here, that's fine. If you want to go outside and visit, you have things to do, that's fine.
Yeah, I think, Annie, you offered to maybe have some slides on your HLA.
I could run through, I might get a bite to eat, but I could run through some sort of, fireside chat on HLA agnostics.
That would be cool.
And that kind of thing.
That'd be fun.
That would be great.
The only bad news is